is currently a student here at Emory also. So I think highly of Emory, otherwise I wouldn't have sent my own kids here. And you'll never encounter my daughter, but she's a theater studies major. That's what's still permissible to exist.
We've talked about the dark side.
Yeah, the dark
business,
side.
business. They're the dark side. It's okay.
But for many years I taught in the neuroscience and behavioral biology major, which is interdepartmental, and I did a core course there. I must have taught that course for 20 years, and then I suddenly discovered that I wanted to do something different. So 4 years ago I switched to intro bio, and I like it. Most of my students in intro bio are first-year students, and first-year students, believe it or not, are quite different from the other years. Because there is a period of adjustment that you'll all go through. I don't care how many courses you had as a high school student, or there is a period of adjustment and all that. So it's proved very interesting to me, and I've really enjoyed it a lot. The first thing I want to do is ask you how many of you have had an advanced high school course in biology? All right. How many have had any high school biology? All right. So there is, in this room, the typical diversity that I find in intro biology in terms of backgrounds. There are a lot of different backgrounds coming in terms of the kinds of science courses that the students have taken as high school students. And that goes for probably for your chemistry background, and also for your math background. So I'd like to say a few introductory remarks because they might be important to you later, which is, probably you've been already told this, that if you want to be successful in introductory biology, you have to be able to handle chemistry. So if you are not simultaneously taking chemistry and biology, or don't have a good high school background in chemistry, it's advisable that if you're only going to take one science course as a first-year student, that it be chemistry, not biology. Okay? Because it really will depend heavily on chemistry. So it's just an important thing to keep in mind. There are many of our students who take both Chem 141 and Bio 141 at the same time. There are many of our students who just take one of them. And we recommend in the biology department that if you're only taking one of them, that it be chemistry. And that you delay your intro bio to your second year. We have a number of second year students in intro bio. So does that make sense to you, or is it? And today I'm going to give you a lecture that's in the middle of my course. It's not the beginning lecture of the course, it's in the middle. So it's going to show you where you're going to have to be in the middle of the semester. And the other thing that's different about the lecture I'm going to give you today is that one of the things we're trying to accomplish here at Emory is to bring more quantitative reasoning into all the science classes that we teach here at Emory. And so today's lecture is going to emphasize quantitative reasoning. So it's going to be a little bit difficult and a little bit different for you maybe, but I hope you'll play along with me, try to answer some of the questions that are put up, and stop me at any time when you don't understand something. So this is kind of a joke title, Determining Genotype from Phenotype. That's what we're going to do today. And the subtext is that if you can do that, you can be a good gambler. And Mendel would have been a good gambler because he understood the laws of probability as they pertain to genetics. Okay? So here's the outline. We're not going to be able to get to the 2 gene crosses, but we're going to do simple Mendelian genetics, how things that are determined by a single gene are transmitted from one generation to another. And then we're going to figure out how you can determine genotype from phenotype with test crosses. Can anyone tell me what genotype is?
The actual genetic makeup.
The actual genetic makeup. So you have that you have. And can anyone tell me what phenotype is?
The physical manifestation of that cause.
That's correct. All right. So we have some, and then we have some basic background understanding that you need. Pedigrees are family histories that track phenotypes. Okay? They track phenotypes. Like, I can track the phenotype of blue eyes in my family. You notice my eyes are brown, right? And I have 2 children with blue eyes and 1 child with brown eyes. My wife has blue eyes. So we could track the phenotype of eye color in my family. And from that, we might be able to infer the genotype. We might, or we might not. Depends on what you do. And then we're going to talk about how different types of genes are inherited in pedigrees. All right, so here's an illustration of the difference between genotype and phenotype. You're a genetic counselor. A newly married couple writes to you because they want to have children. And the woman has a disease called PKU. Has anyone ever read the warning on a Diet Coke can that says phenylketonureacs, don't drink this stuff? If you just pick up a Diet Coke can, it'll say that. And that's PKU, phenylketonuria. It's a disease where you can't metabolize the amino acid phenylalanine. And aspartame is a small peptide that contains the amino acid phenylalanine. And so therefore, you're not supposed to drink it if you have PKU. So you can look that up. I'll show you a picture. So, and if you are exposed as a child to phenylalanine, then you develop severe dementia. Okay? So it's not a pretty disease. And so her PKU was detected at birth, and she thrived on a special diet. She was not fed any phenylalanine. That she stopped in adulthood. You have to inform her about the genetic causes of this disease and the relationship of genotype to phenotype. You also have to advise her and her husband about the risk of passing on this disease and the effect of her disease on her unborn child. So what are the probabilities that she will pass this trait on to her children? That's what she wants to know, right? She comes in, she tells you, she asks you this, you know. It's not like blue eyes and brown eyes, what is the probability of going to pass on blue eyes or brown eyes? Well, that's an important question maybe, but it's not a life or death one, right? So this is more of a life or death type question. So here's the Diet Coke thing. Phenylketonureacs contains phenylalanine, all right? So, and here's an example of the woman here. She's photographed on her wedding day. Here's her sister, who's born 2 years earlier than her. Her parents were unaware that she might have PKU, and so she was served a normal diet of breast milk and formula. And as a result, she is demented for the rest of her life. But once they diagnosed this, then they were very acutely aware of it. And so the next child that was born, they immediately tested for PKU, put her on a special diet. And there she looks today. She's fine. But she has this genetic defect. So does that scare everybody? I think it's very interesting. I think it's, and it shows you the importance of genetic counseling and of genetic tests and what we're doing here. So what happens here is that there is a amino acid called phenylalanine. The phenylalanine is in all of our proteins. It's an essential amino acid of all our proteins. So it's not like you can live without this stuff. You can't live with it, but you can't live with too much if you're a phenylketonuria addict. But you have to have some, otherwise you're in trouble. But if you have too much of it, in a normal person, it's broken down and metabolized. And there are a series of enzymes that are involved in this breakdown. All right? And those enzymes are essential for the breakdown. And it's this enzyme right here that is defective in people who have PKU. So it is not a functional enzyme. And there are a number of mutations in the gene that give rise to that enzyme being non-functional. Okay? And so that's what causes PKU. All right. So now we have to think about what happens to produce gametes in reproduction. Because we're interested in how this mutation can be transmitted from generation to generation. And how is genetic information passed from generation to generation? It's passed through the gametes, right? So females make eggs and males make sperm. The eggs and sperm get together, they produce a zygote. So you get essentially half of your genetic complement from your mother and half of your genetic complement from your father. Now that's not quite true. If you're a male, you got this little piece of crap instead of a second X chromosome. It makes you sort of, not so great, but we can get by with it. I mean, but to a first approximation, it's true. You get half from each. And certainly the females, that's true. So let's consider a mythical organism that has only one type of chromosome. How many types of chromosomes do we have? Does anyone know?
26.
How many?
30.
23. We have 23 different kinds of chromosomes. We get 23 chromosomes from our mother, 23 chromosomes from our father. We have 46 chromosomes in 23 homologous pairs. Okay? That's what they're called, homologous pairs. One from your father, one from your mother. The X chromosome, of course, in females, you get one from your father, one from your mother, but in males, you get X from your mother and Y from your father. Okay, so in this mythical organism, there's only one pair of homologous chromosomes, right? There are such organisms, they're very simple. So before the production of gametes, each chromosome duplicates. Okay, it duplicates. All the DNA is exactly copied. Okay, so that is a true duplication. It's just like putting a piece of paper into a Xerox machine. There's an exact copy made. These two chromosomes, they all have the same genes on them, but they might not have the same alleles. Does anyone know what an allele is? What? Who said? An allele is a different form of a gene. So let's say there's a gene that produces the pigment for eye color, an enzyme that produces eye color. It can exist in two different forms, brown and blue. Okay? It's the DNA that makes up that gene has a certain sequence, and it's slightly different in the two forms of the gene. So that gene makes, one allele makes one protein, and the other makes a slightly different protein. Okay? That's what alleles are. They're slightly different forms of the same gene. Okay? So, in most of us, we all have the same allele for the PKU gene that makes that enzyme. It's exactly the same. And therefore, we always make a good form of the enzyme that works. There are several mutations that can occur in that gene where you form an allele that makes a defective enzyme. Not all alleles are defective. Some can be just slightly different. But in that case, it's defective. It doesn't make an enzyme that works. So therefore, it's not very good. So, but now you have two chromosomes, each duplicated, and this one, they both have the same genes. This one might have different alleles than this one, but there are two copies. The first thing that happens in meiosis is that these two chromosomes come together and then at what's called the metaphase plate, and then they separate like this. And so you separate your maternal and your paternal alleles, right? You separate them. Okay? Now with a one chromosome organism, it's pretty simple. Then these duplicated chromosomes fall apart in the next division of meiosis, and so you get 4 gametes, each of which is now called haploid because it only has one copy of each chromosome. You are what? What are you called? Diploid. Diploid because you have 2 copies. All right? So that's the basics of genetics. So in this case, let's say there's a different allele on the blue chromosome for a particular enzyme than on the red chromosome, right? What is the likelihood that the next generation is going to receive the blue allele versus the red allele? Can anyone tell me? What's the probability? This test says to me that half, let's say this is making sperm because it's in a male, half the sperm are going to be blue, carry the blue allele, half the sperm are going to be carrying the red allele, so that means it's 50%. 50%, 1/2.
Excuse me? Yes. On the cell phone? On the cell phone outside? Sorry.
No problem. Okay, so that's an important idea to keep in mind. The law of segregation. Alleles are separated in meiosis 1. That's this process. There are 2 divisions in meiosis to form gametes. The alleles are separated in meiosis 1. And the law of independent assortment says pairs align independently in meiosis 1. Okay, so what does that mean? We'll see now a little better if we consider an organism that has 2 different chromosomes, right? We have 23, remember? This one has 2. So, normally in genetics, we show alleles by letters. So we say, this person has the big G allele, this chromosome has the big G allele, and this one has the little G allele, okay? For the same gene. So this, and maybe one came from the father, one came from the mother, this from the father, this from the mother. And we say that this person has a genotype of big G, little g, right? Because that defines their genetic composition for that gene, right? And they are called what?
Heterogeneous?
Sort of. Hetero what? High heterozygotes. They're called heterozygotes. If you had both your chromosomes with little g, then you would be called a homozygote. Right? So this person is heterozygous for this organism for the G gene, and heterozygous for the W gene. Right? See that? Okay, so now let's apply the laws of segregation, Mendel's laws, here. So in meiosis 1, we segregate alleles. Homologous chromosomes pair and then separate. Right? But wait a minute, guys. They pair independently of one another. So it's possible for both the blue sets of chromosomes that came from the father to be on one side, and the orange ones up from the mother on the other side. Or it's possible that they go like this, right? It's all a matter of chance. It's all a matter of chance. And this is going to happen 50% of the time, and this is going to happen 50% of the time. That's what's meant by independent assortment. So the different chromosomes assort independently. We're going to separate maternal and paternal alleles, but we're going to separate them for different chromosomes independently. That's really, really important to understand. And this is something that Mendel worked out. So the end result is that you'll get 1/4 gametes that look like this, 1/4 gametes that look like this, 1/4 gametes that look like this, and 1/4 gametes that look like this. And so that means that we have to consider those probabilities when we look at crosses. Now what we're going to do today is we're only going to consider what's called a one-gene cross. So we can ignore all the other chromosomes and what they're doing because we're only going to follow one set of chromosomes, right? We can ignore all the rest because we're not following them. If we did follow them, we would have to take it, and we wanted to account for all the different genes, we'd have to take them into account. Okay, so the tools of transmission genetics are things called Punnett squares, and then probability rules and branch diagrams, and then pedigrees. So let's look at PKU. The genotype is allele equals the version of a gene. If you have PKU, you are homozygous for a mutant allele of phenylalanine hydroxylase gene, sometimes called the PAH gene or the PKU gene. So you are homozygous for that mutant. You have to have count them, 2 bad alleles in order to suffer from the disease. Can anyone give me a reasonable reason why if you only have 1 bad one, you're okay?
Because there's a dominant allele that would surpass your recessive allele, which would be your PKU allele.
Okay, that's absolutely correct, totally correct. I will explain it slightly different. I will say that the PKU allele produces a defective enzyme. So that's on one of your chromosomes. Let's say you're a heterozygous, it's on one of your chromosomes. So you're making a bunch of garbage there, right? On the other chromosome, you have the normal allele, so it's making good enzyme. And it makes enough to get you through the night, right? So you survive. So we say that the normal allele is dominant over the mutant allele, which is absolutely what he said, right? We say it's dominant because if you have one good copy, you're okay. So that's called dominance. We say that the mutant allele is recessive. We say the normal allele is dominant, okay? That's not true for all mutant alleles. Some mutant alleles are dominant. And we say it's an autosomal recessive. So we say it's a recessive, this gene, because it's dominated by the normal allele. And we say it's autosomal because we mean it's not on a sex chromosome. That's all it means. It doesn't mean anything fancy. Just means it's not on a sex chromosome. So you have 22 autosomes, and you have one sex chromosome. The phenotype is mental retardation. So here we go. So here you see a cross between two individuals. One individual has big P, big P. That's the normal allele for this enzyme, right? And it is given a capital letter because it is dominant, okay? It's just standard notation. It doesn't mean anything special. And that person mates with someone who has PKU. If they have PKU, we know they have two recessive alleles, right? We know it with a certainty. And now they're going to get together and have children. And this is the cross. We're going to cross this times this, and all the children will be that. All right? They're heterozygous for this gene. We call them carriers. Why do we call them carriers?
Because they still have the gene, but it's just unexpressed.
That's right. And it can come out again in the next generation. All right? So that's easy. We don't have to do anything. All right. If big P, little p has the same phenotype as big P, then big P is dominant. That makes, that's what just sense. The genotype of all these individuals is big P, little p, right? Everyone agree with that? But the phenotype is expressed as P blank because it's ambiguous from looking at the phenotype. Genotype. We don't know. They could be big P, or they could be big P, little p. You look at the person, you ask whether they can metabolize phenylalanine, you ask if they have a good enzyme. They do. We just don't know unless we do a genetic test. All right, so now we're going to cross them like this. We're going to cross two carriers. So what happened? How were this woman born, these two sisters born that I showed you? They both had normal parents. They both had normal parents. The parents married, they had kids. Both their children have the disease. So both parents must be, they absolutely must be big P, little p. Otherwise they couldn't have children who have little p, which is what they have. So they get together and they mate, and here's what happens. So we do what's called a Punnett square. So the mother is going to contribute gametes that are either like this or they're like this. One or the other, right? If you only have one chromosome that you're following, half your gametes are going to be big P, half are going to be little p. The father does the same thing. So we put them together in all combinations, and that's what you get. That's what's called a Punnett square. It's the simplest way to predict what's going to happen. So 1/4 of the children should be big P, big P. They will look normal. They will be normal. And they will not be carriers, right? 1/2 the children will look normal, and they will be carriers. 1/4 of the children will be little p, they'll have phenotype. All right? Now, this is probability, this is not certainty. So you say, well, they had two children, both of them had PKU, how could that happen? Well, if they had 20,000 children, I would predict that 1/4 of them would be little p, but they would be tired, believe me. Taking care of all those kids would be a big problem. So, if we look at this, then we say, determine the ratio of probability. The genotypic ratio is there's 1 PP to 2 big P to 2 big P little p's to 1 little p little p. That's the genotypic ratio. And the phenotypic ratio is 3 to 1, right? Because all 3 of these boxes are the same phenotype. They're different genotypes, true, same phenotype. Normal or PKU, the normals are here. The PKU is here, right? So now you know the rules of probability, and so you can assign numbers. You say 1/4 this, 1/2 this, 1/4 that. So if you have 2 carriers and they mate, then the probability of them producing carriers is 1/2 the probability of producing a child with little p is 1/4. So this is where gambling comes in, right? If you had a 4-sided coin, I know it's a little hard to imagine, and only one side paid off in gambling, right? You know, there are 4 different colors on it. You throw it up, and only one color gets paid, right? That means a probability of 1/4 of you getting paid, right? Yeah, on occasion. You'll get 2 of them in a row, right? That's like having 2 PKU kids in a row if both parents are carriers. But, you know, on average, that's a bad bet, unless you're getting a good return. Certainly for 1-to-1, it's a terrible bet. Let's say you bet $1 and you get $2 if you win. That's a bad bet. That's a really bad bet. Because 3/4 of the time you're going to lose, and only 1/4 of the time you're going to win, and you're only going to get paid $2 for a win. It's a bad, bad bet. So just don't do it. You know, the games that attract the most attention of the big gamblers in Las Vegas and places like that are ones where the probability of winning or losing is about 50-50. Then it actually makes sense to gamble if you know what you're doing. All right, you ever read James Bond? See Casino Royale? It depends on Baccarat. That's a game of 50-50 probability, essentially. So you're going to have 3/4 normal, 1/4 PKU. That's the idea. So, two-thirds of the individuals who are normal will be carriers. Does everyone see that? See how I'm reasoning from my data? If I take, let's say these people had 30 kids. If I take all the normals from them, 2/3 of them will be carriers. Does that make sense to you? I know they're normal, so they have to be in this group, right? And this tells us that 2 out of 3 will be carriers, right? That's how you reason from your data. So these are where all these numbers come from. So this is the kind of thing we're trying to get you to do. Okay, so here are my 3 children. We talked about their eye color, but the fact is that they're nerds. You know? The nerd phenotype is determined by the nerd gene. I hate to tell you this, and it's dominant. The big N is dominant over little n. Ron and Christine are nerds. We're both nerds. My wife is an English professor, I'm a biology professor. You know we're both nerds. And have 3 nerd children. All 3 of our children are nerds. And what I want to know are, what are my genotype and my wife's genotypes? What are they? Can you tell me? from my children. I'm a nerd. My wife's a nerd. All three of my children are a nerd. What's my genotype? What?
You can't tell.
You can't tell. You can't tell. You absolutely cannot tell which is the case. Here, let's say that I'm big N, little n. And let's say my wife is big N, little n. Right? Let's do the Punnett here, right? What are we going to get? We're going to get then we're going to get. So the probability is that only 1/4 of my children will be not nerds, right? So whereas 3/4 of my children, each time we have a meeting, there's a 3/4 probability that they'll be a nerd, right? So I have 3 children, so it's 3/4 times 3/4 times 3/4 that they'll all be nerds. And that's not so bad. It's 3 times 3 times 3 is 27. 4 times 4 times 4 is 64, so it's 27/64ths that all 3 of them will be nerds. So the only way to really tell is for me to have a lot of children, see if a non-nerd pops up. Every once in a while, you have to do a lot of crosses, but you know, Christine didn't want to do that to figure it out. But there's a simpler way, and she was even less likely to consent to this, which is, how can we find out? You can find out by taking me and test crossing me to a non-nerd. But she didn't want that. I have no idea why. I tried to convince her, but I said, we've got to determine whether I'm heterozygous or homozygous. I said, we can do the same for you. So you have to mate me to someone who is not a nerd. We know that person is little n. We don't know what I am, but if I mate to them, I'll know right away whether, pretty much, after a few kids anyway, whether my genotype is big N, big N. If I'm big N, and I mate to this person, all my children will be nerds. Every one of them. Every single one. Right? Because they're all going to get a big N from me. No matter what they get from their mother, it doesn't matter. Because I will determine their phenotype. Not their genotype, but their phenotype. But if I'm big N, little n, then I expect half the children from that mating to be normal. Let's do it. If I'm big N, and I mate with a non-nerd, then half the children will be big N, little n, and half the children will be little n, little n. So that's how we could determine it. But again, you know, it just didn't happen. So, but we can do this with genetic organisms. And Mendel did it with peas. He liked peas. He worked with peas. You can do it with fruit flies. You know. So here we go. We have a gene, little r, that causes wrinkled seeds. The big R version of it is a round pea, and big R is dominant over little r. So if we test cross an unknown round seed with a wrinkled seed, which must be homozygous recessive, then all the P's will be round. That proves that this P was homozygous. Here's another gene. It's the yellow gene. Yellow is dominant over green in P's. You didn't know that, did you? Isn't that weird? Yellow is the dominant color of P's in P's. So, but let's say we have a yellow individual. We don't know whether it's big Y, or big Y, little y. But we cross it and we find that half of the P's that are produced are this and half are this. That proves that that guy was a heterozygote, right? Does everyone agree with that? All right. So now you're learning laws of probability. So this is just saying what I said. Laws of probability. So this is just saying what I said. And now we'll do a little probability game. So here's a dice, right? So now you're going to gamble. I teach everybody how to play craps, but we don't have enough time how to play craps, but we'll get the basics down here. All right, so there are 6 numbers on a die, right? Exactly 6. And each one has an equal probability of appearing if it's a good die, right? So, I mean, you can load die. Unloaded die, this is what you can expect, right? So what's the probability of rolling 1? 1/6, right? Now, if I say and, then multiply. All right, just remember that rule. So, Justina rolls 2 dice. What is the probability of her rolling boxcars or cornrows? That's common parlance in craps for 2 6s. Okay? It's called boxcars or cornrows. So, what is the probability of getting 2 6s in a row? You know what the probability of getting 1 6 is, right? What is it?
1/6.
1/6. What's the probability of getting 2 6s in a row?
1/36.
1/36. So this is true if you play Monopoly or any game like that where you throw 2 dice simultaneously. Doesn't matter whether I throw 1 dice and then throw it a second time or whether I throw 2 at the same time. Doesn't matter. They're independent of that dice, so it's 1 in 36. So you only have 1 in 36 chance of throwing a boxcar. Now, there it is. There's boxcars. Also, of course, cornrows. I don't understand where they get boxcars from it. But cornrows make sense because it looks kind of like that. All right, or then add. What is the probability that your Tunde will roll either a 6 or a 1 with 1 dice? 1 die. I have 1 die, and I ask, what is, I'll pay you if you roll either a 6 or a 1. What's her probability?
2 out of 6.
2, 6. Everybody says 2, 6, right? Because it's 1/6 that she'll get a 6, 1/6 she'll get a 1. Add them together. That's 2, 6. We could reduce the fraction to 1/3. It doesn't matter. 2, 6, 1/3. On a test, they're the same. If Justin rolls a 6 first with 1 die, I'll roll a 6 first. What is the probability of the next roll being 1?
1/6.
Everybody agree with that?
Mm-hmm.
If I roll, I have a die here, I throw it, I get a 6. And I pick it up, and I throw it again. What's my probability of getting a 1?
1/6.
It's 1/6 again, because they're independent events. That's like 2 matings in the same family, right? 2 matings in the same family, right? So you can think about my eye colors now. We have a little bit more information there because I did do a test cross with my wife. She is blue-eyed, so she's little b, right? And I have 3 children. 1 of them has brown eyes, 2 have blue. What is my genotype? I'm a heterozygote, right? Have to be, right? I have to be. And my two blue-eyed children, what's their genotype?
Recessive.
They're both little b's, right? And what is the genotype of my brown-eyed child? Big B, little b, right? Has to be big B, little b. So they, you know, because they're independent. They're independent. The two children, all three children are determined independently. The sum of all probabilities describing a single event is 1. That's something, a really important thing to consider. If I ask you, what is my probability of having either a brown-eyed child or a blue-eyed child? 1. You know, it's got to be one or the other, so the sum of the probability is 1. 100% will be either one or the other. All right. What is the chance of Yak Ting rolling anything but a 6 using 1 die?
5 out of 6.
What?
5 out of 6.
Yeah, that's correct. But how did you get it?
Well, since your probability is 1/6 of rolling a 6, and your entire probability has to equal 1, you just subtract 1/6 from 1 and you get 5/6.
Does that, you disagree with that?
Oh, no.
You think that's good?
Yeah.
You would give him like full credit on that question?
Yeah, I think so.
Well, that's good. I'm glad you're a fair teacher. All right. All right, if Channy rolls 1 die twice, what is the probability they will be different numbers? Now think a little bit harder. Your eyes are swimming back in your head. You got any guess about how you do it? So what's the probability you'll get some number on the first roll?
1/6.
1. Some number. You're going to get some number on the first roll. It's 1, right? Now, the next number you have to get can be any of 5, not the one you just rolled.
So would it be 5 out of 36?
So you have, the first one is going to occur, right? So that's like me asking you, what's the probability that a 5, you won't roll a 5 on the next turn, right? So it's 5/6, right? It's the same question as the one above. All right, very good. So we go back to this, and everything is done by the same laws of probability. So we can say that in a monohybrid cross, is what we call this, a monohybrid cross, because both are heterozygotes, and we're only following one gene on one chromosome, then the genotypic ratio, probabilities are 1/4, 1/2, 1/4, and the phenotypic probabilities are 3/4 and 1/2. And of the normal individuals in a dominant recessive relationship, 2/3 will be carriers. All right. So maybe we'll skip this one. Or are you enjoying this at all?
Yes.
All right. So the only one person is enjoying it. If you're enjoying this at all, we'll do these. So here we go. You're a geneticist, and a lot of geneticists like to use fruit flies. If you do some research in the biology department, which you might do over the course of your 4 years, we have a number of students who do research and get credit for it, so it's very important. Then you might do Drosophila. So you have one pure breeding brown-eyed strain of flies. So every fly, no matter how many times you mate them with one another in the population, always has brown eyes. And one pure breeding strain with red eyes. And it turns out that the red-eyed ones are recessive to the brown-eyed ones. So we say that brown is a dominant allele, okay? But we know they're both homozygous strains. Now we put them together and we mate a female red eye with a male brown eye, or vice versa. It doesn't matter, male or female, because it's an autosomal trait. And now we expect 100% of the next generation to be brown eye. Does everyone agree with that? 100%. All right. Now we're going to take those big B, little b flies, because that's what they have to be, right? From one parent they got a little b more. And we're going to mate them. And that's the monohybrid cross. And now, thinking with your probabilities that you had from the last thing, if a geneticist picks 4 flies at random from the F2, what is the probability of getting all brown-eyed flies? Well, first of all, what's the probability of getting a brown-eyed fly? Does anyone know? Out of this cross. You know, no matter, you just look for a brown-eyed fly. What's the probability of getting a brown-eyed fly? Let's go back to our punnets, because some of you are, this is like crossing big B, little b, right? It's the same with P, right? P, B, I don't care. So we expect what percentage of the flies to be brown-eyed, the dominant? 2/3, right? 2/3, everyone to go with this? 2/3? 2/3, no one agrees. Back there, you don't agree. Why not? Because I'm going to cross a big B, little b with a big B, little b. So to get all of these people, all of these flies will be brown-eyed, right? Every one of them. So the probability should be just exactly what my doubter back there said, 3/4. Right? Not 2/3, but 3/4. See, she stuck to her guns. She looked at me like I was a criminal. And she knew I was making a mistake, and she stuck to it. And so, yeah, you're right. So 3/4 is exactly what we can expect. And here we go. Remember 3/4 from back here? It's the same thing. So what is the probability that if I just at random pick out of the population 3 flies, that they will all be brown-eyed? The probability of picking the first one is 3/4, right? Probability of picking the second one is 3/4. Probability of picking the third one is 3/4. So it's again 27/64, right? Okay, that's good. Should be, I hope so. Oh, it's 4, so it's 81/256. So I'm sorry, I said 4 flies, not 3. It's 3/4 times 3/4 times 3/4 times 3/4, or 81/256. What's the probability of all red-eyed flies?
1 out of 256.
So it's 1/4 times 1/4 times 1/4 times 1/4. All right? Good. 1 and 256. And 1 brown-eyed fly and 3 red-eyed flies. This is really hard. Now you've answered too many of them. Let's have someone else try to answer this. This is a hard one.
Is it 3/4?
It is, all right, so you would, this is a really good start, but it's only a start to the answer. So your solution is that 1/4 times 3/4 times 3/4 times 3/4. No. It's the other way around, of course. Absolutely, I'm an idiot. You know, you got to stick with me here, you know. Right. So that's your solution. And I agree that that's a potential solution. So the first time I reach in, I get a brown-eyed fly. Then the second time I reach in, I get a red, then the third, a red, a third, a red. But I say, no, no. What if the first time I go in, I get a red-eyed fly, right? But the second time I go in, then I get my brown-eyed fly. Then I get a red-eyed fly. Then I get a red-eyed fly. So there are all these different possibilities, right? In fact, there are 4 different ways I can do that. Just like in, you know that the probability is 50-50 of having a boy or a girl, right? But families, you can have families in different sequences. I have boy, boy, girl, but it's just as likely to have girl, boy, boy, right? So you have to consider all the different ways you can do it. And there are 4 different ways. So it's 4 times what you said. I think that's true. If I wanted, yep, that's what it is. 4 times that, right? Because there are 4 different ways of getting it. You can see that because I can pick the brown-eyed fly either the first time, the second time, the third time, or the 4th time. So there are 4 different ways. So that was great. Thank you for sticking your neck out and getting us there. So we'll go through here. So in pedigrees, we can show carriers by showing them as being half one color and half another. But normally in pedigrees we don't do that. You have to figure out what the genotype is. We just show the phenotype. And males are always boxes and females are always circles, and I don't know why. And then we number the generations with Roman numerals. So here you see a pedigree. From this pedigree, if you're solid colored, then that means you have the defect. So looking at this, can you tell me what kind of gene causes, what kind of allele causes this defect? Is it a dominant allele or a recessive allele? Somebody hasn't answered the question. Is it a dominant allele or a recessive allele that causes the blue trait? That's correct. It's a recessive one. All right. Now this is easy to figure out because, first of all, you see that there are carriers. All right. Normally we have to deduce that. All right. So here is a real pedigree. A real pedigree. We see in this family a affected individual, right? They have, you know, some genetic defect. PKU, cystic fibrosis, you know, whatever. They're getting together and over the generations, the next generation, their kids, they mate with a normal individual, their kids are all normal. Then there's another family that marries in. You see there's another family that came in and married in, and everybody was normal in that family. And then all of a sudden it reappears again. The P usually means the proband. That's the first person who's born who's noted to have the defect, and then you look back to see who had it in the past. This means twins. It just means twins. It's nothing special. So that is the hallmark of the autosomal recessive, because both males and females have it, and you know all of these guys must be carriers, right? Because their mother had the defect, right? So she was homozygous. So all of these are carriers, and they must have married from a family of carriers. This person must be a carrier, right? If they weren't, then there's no possibility of this appearing. So we can say definitively that all of these are heterozygotes for the trait, and that this person is heterozygote. Now let's look at an autosomal dominant. Here you see the mother again has the trait. In the next generation, the trait appears immediately, even though she mates with a normal male. And in the next generation, it appears even again, though there's mating. Whenever there's an affected individual, there are affected children. Because half the time, you'll pass on the dominant allele which causes the disease. Affected individuals all have an affected parent. Skips generations, right? Skips generations. Recessives skip generations. So here's a albinism, a rare recessive allele, and these two individuals don't have albinism, but they get together, and then their children don't have albinism. They marry out. People marrying into a family of a rare genetic disease are usually assumed to be normal. So you can almost assume that their genotype is big A, big A, big A, big A. But now we get down here, and this is not always a good thing to do. You shouldn't marry your cousin, especially if you know there's a recessive genetic mutation in your family. You shouldn't marry your cousin. And now, in the next generation, appears albinism, right? And so, reasoning back, we reason that these guys must be both carriers, right? They have to be be normal and to generate children who are albino. And so you can reason back from them, and then you must be able to, somewhere, they must have gotten that little a. So they had to have gotten it from this parent, right? And somewhere this guy must have gotten their little a, so they must have gotten it from this parent. And ultimately, one of these two parents back here must have it. All right? At least one. Now here's the last question, and then I'm going to let you go. Here it is. Now, I'm going to tell you the truth. My middle child graduated from here from Emory. Now he's in medical school. He had to pass his medical boards, Step 1 boards. This is a question from the Step 1 boards. All right? So this is what they consider important to be able to do in medical school. I'm going to give you a lot more information than they gave him. First of all, cystic fibrosis. Now, the medical student is expected to know that cystic fibrosis is caused by a recessive, autosomal recessive. Okay? You have to have 2 of little cf genes to get cystic fibrosis. So you're supposed to know that. All right, I'm going to tell you that. That's just something you memorize. That's what medical students do. They memorize things. We're going to use that data with what we learned today to figure out the intellectually interesting part of this problem. So here it is. A man with cystic fibrosis. We know that, what's that person's genotype? Little cf, right? That's what you're saying, right? Little cf. We know. Conceives a child with an unaffected woman. It's possible, yes, that can happen. There was a cystic fibrosis person who marries an unaffected woman. He is concerned that the child will suffer from cystic fibrosis. based on the pedigree below, what are the chances that the child will be affected? All right? So here is the man we're talking about, right? This is the man we're talking about. He has cystic fibrosis. Here's the woman he marries, right? And here's the child. We don't know what sex it's going to be. It doesn't matter, but we don't know what sex it's going to be. We're genetically counseling. But look at the woman's family. What is this telling us about the woman's family?
They're carriers.
These parents, her parents, were both carriers, right? Doesn't it tell you that? It automatically tells you that both her parents were carriers. Because otherwise they could have never conceived a child that is homozygous, little cf, if they themselves are normal. Right? So we know that genotype. Great. We know this person's genotype. Right? So all we have to do is calculate what is the probability of this person's genotype being big CF, little CF, right? Because if she's big CF, the probability of this child being affected is what? 0. 0. 0, right?
Yes.
So we just have to calculate that probability, and then we have to calculate the probability that it will pass on to the next kid, to the next generation. So we'll do that. So the first thing you do is put in all the genotypes. So plus is often used to mean the big letter, okay? It just means the normal, right? So it's just a, so instead of writing that the genotype is little cf, big CF, you write it's little cf plus, okay? Because there's a good allele. So we know the genotype of the parents here must be this. We know the genotype of these parents must be this too, right? Because otherwise they wouldn't have having a kid that had cystic fibrosis, right?
Yeah.
So we know those genotypes. We know this person's genotype. That's absolutely certain. And we know this person's genotype. Now we have to figure out the rest. All right. The probability that this child will receive the defective allele from his father is 1. Right? Does everyone agree with that?
Yes.
It's 1. So we know one number in our little equation. It's 1. Because that is the probability that the mutant allele will be passed on. 1 times something. So that we know with certainty. What is the probability that this person is a carrier? I say it's 2/3. Now, you say it's 3/4, right?
No.
Yes, that's right. So that's why it's, yeah. You see, you're thinking. Good. That's excellent. So what she's saying is, we know that this is not little cf, right? So it's either big CF, little CF, or big CF, right? And of those, it's twice as likely that it'll be the heterozygote or the carrier than the homozygote. Let's do it. In this mating here, right? Let's do that mating. Let's do that mating. I can't fool you back there. Whoever you are with the Emery shirt. That's good. All right, so we've got little CF, big CF, and little CF, big CF. All right, so you know that this person is not that. But then there's big CF, little CF, and big CF, big CF, and big CF, little CF. Right? So of the, there's no other color? Darn it. Of the non-affected individuals, which are these, right? Two-thirds are carriers, right?
Yes.
So there's a 2/3 probability that she's a carrier. Great. So now we have another number in here. 2/3 probability that she's a carrier. Now, probability she will that this child will receive the little CF from a mother, if she is a carrier, is what? 1/2, right?
Mm-hmm.
Because half the time she'll pass it on and half she won't. So now, you just multiply all that together, and you get 1/3. So you can go and tell these parents that there's a 1/3 probability, that their child will have cystic fibrosis. Do they want to take the chance? Okay? The other 2/3 of their children, what will be their genotype? So they'll be carriers, right? They'll be carriers. Okay. So that's enough for today, but I hope that I both scared you a little and challenged you a little. And actually, as I say, this is the middle of the course, but this is what I do, you know, and all of us do this. And we have questions like this appearing on our exams. And I greatly appreciated your willingness to participate and answer questions. And there was one important lesson that we learned, which is that the teacher makes mistakes. And when you see the teacher making mistakes, you raise your hand and you tell them that he's making a mistake, because he'll appreciate it, or she'll appreciate it. Because we all know that we misspeak. And we all know, so that was good. And I'm very glad, you know, because it's easy to not think on your feet all the time. So that's good. Never, if you really think you're right and the instructor is wrong, then ask the question. He will appreciate it, she will appreciate it, regardless of whether he's wrong or right. Okay, so good lesson to learn in life. So any questions from you all? That's a... Those of you who aren't from the South, that's singular. If it's plural, it's all you all, right? So I learned that when I moved here from New York. So any questions about anything? I mean, biology department, bio major, NDV major.
When you're teaching Bio 1 and first year biology, would you get further into different, like a third or more working in with genetics, or would you have to take an upper level genetics class for that, like eye traits or hair traits?
Well, what we do in the first semester is we do simple Mendelian genetics up to tracing 2 genes simultaneously. And then in the second semester, there's a lot of work on gene expression and the actual structure of the genome. So our sequence is very heavily biased towards cell and molecular biology. If you want advanced genetics, right, many of the bio majors take as an elective course a course called human genetics. And that's at the 300 level, but You know, advanced students are taking it in their sophomore year, and other students in their junior year. Then we have a course on epigenetics, which is also very popular, and that's mainly seniors who take that. And it's an extremely good course.
How many biology and immunology classes does Emory really offer?
All right, we have no courses on virology, and we have no courses on immunology. There is some immunology in the intro sequence, but we currently do not have a course on immunology. But we hope to soon. We've had one for the last 8 years, but the person who taught it is leaving. What?
Is that why it's still on the course atlas?
Yep, it is. Because the person who taught it for the last several years is an immunology course. It's on the books, but won't be taught this year. Probably not next year, but the following year will be taught. So when you're a junior and ready to take it, it'll be there. Now, if you really, really want to do immunology, you can take the graduate course in immunology. A lot of people do take graduate courses. To get an honors thesis, to do honors, you must take a graduate course. So, nope, you don't want to do that.
Sounds like scary.
Yeah. Well, when you're a senior, it doesn't sound so scary.
Yeah, it's easier then.
It's easier then.
Yeah.
But, yeah. So there is a course that is how to describe it. It's not the molecular biology of immunology, but it's the epidemiology of immunology. That course is still offered. So there are two sort of areas. There's the molecular biology, what are antibodies, how are they formed, and on a molecular level, and then more, there's more of a course that is currently taught that's about how do pathogens interact with the immune system to co-evolve. Okay? So that course is still offered to undergraduates. And that's by Antioch. You can look that up. The one by Schlenke is not offered. Next year, the one by Antioch is.
What made you decide to stop teaching NBB and switch to biology?
Well, when you teach a course every year for 20 years, and you teach it both at the undergraduate level and the graduate level, you kind of say, it's time for a change. And although I passionately love neuroscience, and all my research in my lab is on neuroscience, I just wanted to do something different. And so I decided to teach intro bio. And I'll probably do this coming year and maybe another year more, and then I'll probably switch again. But I'm not sure where. It depends on what the department wants me to do. I'm a little bit more flexible than other people in the department because I'm a little older and I don't have any responsibilities at home, so I can change courses without too much trouble to my life. And they move me around in time slots too, you know. Sometimes I get 8:30. Next year I'm at 1 o'clock. I never taught at 1 o'clock. I wonder what that's going to be like. And 1 to 2:15. We'll see what that's like. But they move me around. And Ronald do anything is what I say.
So what I look at?
Any other questions? So all of you are going to be here as freshmen starting the end of August, right? And this is just sort of a summer warm-up, right? So who of you intend to take intro bio? All right, so 3. And who intend to take intro chemistry? All right. So is everyone likely to be a science major?
Yes.
Pretty much.
They're all intending to be science majors.
Okay.
For right now, at least.
And if you're a neuroscience and behavioral biology major or a biology major, it doesn't matter which one, you're required to take both intro chemistry and